Hi, everyone, and welcome back. Next, we are going to hear from Dr. Wolfe. He is the professor and chairman of the Department of Neurology at University at Buffalo Jacobs School of Medicine and Biomedical Sciences, SUNY, where he holds the Irvin and Rosemary Smith Chair. He attended medical school at the University of Texas Southwestern Medical School and trained as a neurology resident and neuromuscular electromyography fellow at the University of Pennsylvania Medical Center. So I will go ahead and turn it over to you. You're going to have a great presentation and probably a lot of questions, so I'll let you get to it.
Okay, thanks. And I hope most of you, if not all of you, can hear me well. And let me get the slides up. Okay, let me go to full screen. All right, I guess, yeah, there we go. Okay.
Perfect. Thank you.
Those are just some pictures of Buffalo. Okay, which looks nice in the summertime. These are my disclosures. And by the way, I thought Dr. Sakdev gave a great talk. I really enjoyed it, and he's helped me, which is good because to be honest, I'm a little bit tired because the International Congress of Neuromuscular Disease is going on right now too. And it was supposed to be in Spain a few weeks ago, and they're sticking to European time. So the sessions are starting at 3 in the morning. In fact, I have to give a talk I have to be on again at 3:15 this morning, and I'm not sure I'm going to sleep before then. So anyway, but I'm glad because it was a great introduction. I thought it was great. And what I'm going to try to do is just supplement what his talk was about. But a lot of these companies he already mentioned, but I've been involved with as well. And he already mentioned the disclosure, so I won't go through it anymore as far as off-label use. Okay, so, let's talk a little bit. The pandemic, it's been tough. But one thing that has been an outgrowth of the pandemic is I think a lot of people are learning new skills or new knowledge. And so I'm going to challenge, I like talking to patients and being involved with these kinds of forums. This is your challenge. If you don't know what your antibody status is, you should know. Some of you I know because I can tell from the chats. But you need to learn about the disease and learn about your antibodies, what you have, what if you don't have it, and so forth. And we're going to talk about antibodies. So that's the leap into that. And then we're going to get to the clinical trials. But we better get going. So this acetylcholine receptor that you guys have heard about and that was drawn very nicely on that simple schematic, it actually has several components. And what I'll mention here is these alpha-1 components, those make up the main immune antigen, the main target of the acetylcholine receptor antibodies is to alpha-1 here, just so you know that. Now this receptor, it is clustered so you can have efficient signal between the nerve and the muscle by rhapsin. To get rhapsin to do that, there's a whole mechanism in place that's largely based on what we call phosphorylation through kinase enzymes. But what they do is lead to sort of this chemical cascade of reactions that allows for this congregation of receptors where they should be on the muscle end plate. We now know that there are antibodies to many of these different proteins that are involved with that cascade, okay? Whether it's agrin or whether it's LRP4, which used to be called MASK, but it's been renamed LRP4, to MUSK, and so forth. We're going to go through some of those, okay? So let's start with the receptor antibodies because this is the most common antibody in MG. And the ones, if you know about your result, it probably was a binding assay. That's the one that's most commonly done. It's measured in nanomoles per liter. And you can see here, for generalized MG who have more than just eye problems, really 90% of you are going to have the antibody. That takes care of, the vast majority of generalized disease. Not all, but the majority, okay? A large majority. For ocular MG, it's lower. It's somewhere between 50 and 70%. If you look at different series. This has to do with specificity. False positives, where you get the antibody, but you really don't have myasthenia gravis, that does occur. It's pretty rare. Usually those levels are very low. I know you've heard about false positives also and false negatives now because of the COVID and the testing for COVID-19. But anyway, there's also modulating and blocking antibodies, okay? These are all IgG antibodies. They're mostly IgG1 with some IgG3. Anyway, some patients with MG, but not very many, will only have modulating antibodies. They don't have the binding antibodies. So when I order antibodies and somebody asked about a panel, I can get these two together. I order them together all the time, okay? The modulating, because 3 to 4% of MG cases who are not positive for the binding antibodies at the top here are positive for those. Blocking antibodies really don't help us very much. There really is a rare case of MG that have only blocking antibodies. Okay. I want to talk a little bit about cell-based assays. So most of the time, pretty much all the time that you have routine antibody testing, it does not involve a cellular system where you're actually attaching the target of the antibody onto a cell. They're done in plastic columns, or they're done on pitted plates, like ELISA techniques and so forth. It's kind of an artificial setup, but there are cell-based assays that have come into development for myasthenia gravis. They're not really they really aren't commercially available at this point yet, but they can be done. My friends at Oxford do them when I order them. I don't order them very often, but occasionally I do. So here you express the antigen on the cell surface of a cell, and human embryonic kidney cells are commonly used. And so it has certain advantages because you can cluster the antigen better, make it more of sort of a natural environment for the antibody. And then you can also set up parallel assays looking at one antigen after the other. And for instance, Oxford does that. They can do this for acetylcholine receptor antibodies, MuSK antibodies, and so forth. Anyway, the point I want to make is a lot of seronegatives out there probably do have these acetylcholine receptor antibodies that are only going to get picked up with cell-based assays, and that's been shown. So up to 60% of generalized seronegative MG patients have antibodies using the cell-based assay for the acetylcholine receptor, and it's up to 50% of ocular seronegative MG patients. And so this is work that's been done in the last decade or so. Okay, now let's go on to some other antibodies, and I just want to blow up the picture of the schematic that Dr. Sakdev showed, for instance. And so again, I mentioned Rapsin, and Rapsin allows the clustering of acetylcholine receptors. So going from this to this. This is outside the muscle cell up here, and at the bottom is inside the muscle cell. There is a protein called LRP4, okay, and we can check for antibodies to LRP4, but it is the receptor for a molecule called agrin that comes from the nerve, interacts with LRP4, and then through enzymatic reactions I mentioned before, there's conformational changes involving MuSK that leads to this cascade of events, okay, through kinase activation. That's what the K stands for down here, is kinases, to allow clustering of acetylcholine receptors. We have antibodies now to agrin here. We have antibodies now to LRP4. We've had antibodies to MuSK since I think 2001, so going on 2 decades. Those have all been linked to myasthenia gravis, somewhat more MuSK more strongly, MuSK and LRP4 more strongly than the others, but anyway, they've been linked. All right, so let's talk about LRP4 antibodies. These are IgG1, okay, whereas MuSK antibodies are primarily IgG4, but LRP4 antibodies are IgG1, and they inhibit that agrin LRP4 interaction, so it inhibits that cascade of events, and if you don't have that cascade of events, you can imagine you won't have proper structure on your muscle membrane to have good neuromuscular transmission. These are not common, except for this one series where half of double seronegative patients, that means patients who are both negative for receptor antibodies and for MuSK. 46% of them were positive, but the other series, you can see the frequency is pretty low, but it's there, of double seronegative patients. And it is estimated, it's somewhere probably around 1 to 2% of all myasthenics are going to have these antibodies. CBA stands for cell-based assays, by the way. IP is immunofluorescence, okay, or immunoprecipitation. So again, 1 to 3% of all MG patients. What do they look like? They seem to be milder to moderate. They can have limb weakness. They can have weakness, you know, of swallowing and speaking and so forth. They don't seem to have thymic abnormalities. So they seem to share that with MuSK. Now, Dr. Sakdev did mention he's had an LRP4 patient who did respond to thymectomy. There are no golden rules. There's no absolutes with this. There just isn't. when you're talking about other antibodies with thymectomy? I know that question is probably going to come up, but we just cannot be definitive. But anyway, the thymus does not seem to be very involved with this form of myasthenia. It is female predominant. Now, the other thing is, except for MuSK antibodies, you have to realize it is pretty common now to see some of these newer antibodies coexisting with acetylcholine receptor antibodies, okay? It does happen, and I'll show you some numbers on that. These patients tend to respond to conventional therapy pretty well and to acetylcholinesterase inhibition. What is that? Acetylcholinesterase inhibition is mestinon pyridostigmine, okay? But these seem to respond pretty well to prednisone, azathioprine, mycophenolate. Oh, and going back to another question that came up in the prior talk. So Dr. Sector, that's my understanding too of why it takes so long for these steroid-sparing agents to work. Look at mycophenolate also. Azathioprine was mentioned, but look what does mycophenolate do? It inhibits an enzyme called inosine monophosphate, IMP dehydrogenase. I think I have that right. But anyway, I just call it IMP for short. Anyway, what does that do? It prevents purine synthesis. What are purines? There's a lot of different purines, but a couple purines you may have heard about before, I'm pretty sure this is right, are adenine and guanine. What are adenine and guanine? That's the A and the G in DNA, okay? You know, we have T and U and A and G, the different molecules that make up DNA, RNA, and so forth. But what it's doing is it's preventing DNA formation, so that way you cannot divide cells and make new cells. So again, it takes a long time to see an impact if the therapy is primarily one that reduces the number of cell divisions and cell replication. It's just going to take a while. So mycophenolate, it's the same. It's the same kind of basic idea. All right. How about the specificity for LRP4 antibodies? It is seen in some other autoimmune diseases. Also, this antibody and some of the other antibodies that have now been described are seen sometimes in ALS, all right, in Lou Gehrig's disease. About 15 to 23% of ALS patients have these antibodies actually in these two fairly recent papers. Okay. Agrin antibodies have come up. Oh, here on the top is a more complex schematic of that interaction of the receptor antibodies. Now they're 5 instead of the 3 before. But anyway, it's just a schematic. You see RAPs in there, which is that scaffold that clusters them. And here you can see agrin coming from the nerve, which would be at the top above my screen and your screen, interacting with LRP4 and all of these little stars or phosphorylation events, okay? That allows for this proper formation of acetylcholine receptor densities on the muscle membrane. Okay, agrin antibodies. These seem to be relatively common, okay? Somewhere around 10%. You can see it here, 15% of 161 MG patients who had some other antibodies and 73 who were seronegative in this British study did have agrin antibodies. The rate here in this study was about 10%. That's what the number seems to be. Again, you can see coexisting antibodies. So of these 5, 4 had MuSK antibodies. One of those also had LRP4, and one had acetylcholine receptor antibodies as well in this series. So you can see these different antibodies together that may play a role in whether patients may respond to thymectomy if they also have acetylcholine receptor antibodies and so forth like that. Okay. Again, agrin antibodies are seen in some ALS patients. It was 14%, similar to that rate of those LRP4 antibodies I mentioned before. Then there's cortactin antibodies. These are present probably closer to 5%, but anywhere from 5, maybe up to 25% of patients with myasthenia gravis, okay? It's also seen in healthy controls. This is cortactin. It shouldn't be confused with contactin antibodies, which fall into the immune-mediated acquired neuropathy field. Certain subtypes of what we call CIDP have contactin antibodies. These are cortactin antibodies, okay? And what does cortactin do? It regulates also through these mechanisms that you can see here, the proper aggregation of acetylcholine receptors. And so again, if you have antibodies to LRP4 or agrin or MuSK or cortactin, you could potentially impair this proper formation of acetylcholine receptor scaffolding and density on the postsynaptic, on the muscle membrane. Here's the nerve up here, the muscle below. Okay. How about other antibodies? Collagen Q has been described, seemed to be quite rare. Collagen 13 was seen in 5 of 70 MG patients, all young women, some who also had low levels of acetylcholine receptor antibodies. But what I want you to realize is, as far as really having a good feel that these antibodies are causative of myasthenia and aren't just a bystander effect where you damage the muscle membrane, expose certain antigens, and then your immune system responds to those. So they're not really disease-causing antibodies, but where we have pretty good data or very good data that the antibodies actually cause disease, that's really just true for acetylcholine receptor antibodies, MuSK antibodies, and LRP4 antibodies. All right. So this is just a chart that I made up when I was asked to a year or two ago to try to summarize these different antibodies. Here you can see the receptor antibodies, and oftentimes receptor antibody disease is divided by age of onset. At a higher age of receptor antibodies, you tend to see atrophy or thymoma of the thymus. In younger age groups, you see hyperplasia. Thymectomy could be implied as a treatment for both of these groups, especially, and it must be if you have an actual thymoma. MuSK is relatively rare compared to the proportion of patients, as are all the rest of them except for agrins here at around 10%, okay? MuSK is 3 to 5%. Rituximab may be a particularly good therapy. Dr. Sakdev mentioned it. It's an anti-B cell through CD20. And for whatever reason, those B cell therapies seem to work very well with disease that's caused by IgG4 antibodies. And sure enough, MuSK is primarily an IgG4 antibody. Here's LRP4. Again, they tend to be milder. They tend to respond to conventional therapy quite well. Agrin, more moderate generalized MG disease. These last few, the actual implications for therapy are not really that clear, okay? But the thymus, except for receptor antibodies, seems to not be playing as important a role. That's pretty well worked out for MuSK. That's what we think for these other antibodies, at least so far, okay? Now, seronegatives, I put variable for the thymus. Again, many of these seronegatives could respond to thymectomy, maybe because some of them really do have receptor antibodies through that cell-based assay I mentioned before. Okay, so let's see, how are we doing with time? We've got about 20 more minutes. What I'm going to try to do is just show pieces of data on therapy that has already been discussed to some degree. And so here is our B cell that was, really nicely described on the prior talk. And I'm going to concentrate on rituximab. Belimumab also reduces B cell counts, but I can tell you the trial that was done, that by the way included MuSK antibody patients as well, it failed. For whatever reason, that trial failed. And rituximab, there's a lot of data like this data showing that it works, but the formal trial showed futility, at least as far as we could tell at a year, for rituximab reducing prednisone requirements. Anyway. So this is some of the rituximab data that came out now 8 years ago. Initially, there were case reports and there were some case series. What this case series showed is this regimen of rituximab in both receptor antibody positive and MuSK antibody positive patients really led to improvement in just about every single one except one patient with receptor antibody disease. And they were followed for a long period of time and were continuing to do well for 2 years to 3 years. They did well. All this excitement from that type of data and other data for rituximab led to a formal NIH, so our tax dollars, supported trial through the NeuroNext network that Dr. Novak ran at Yale. And the formal paper's not out yet, but it should be out soon. But it involved patients who were requiring certain levels of prednisone, but still had active disease. They had to be receptor antibody positive. So no MuSK patients, I think, unfortunately, were enrolled. I would have liked to have seen MuSK patients in this trial too, but the decision was made to be very pure and only include receptor antibody positive patients. Why? It helps with statistics and so forth, that you're not mixing populations and so forth. Anyway, the primary thing that they wanted to see was, would rituximab at basically a year reduce steroid requirements, and it didn't. And there's several reasons why maybe it didn't. It was pretty loose on the ability to reduce prednisone. And when patients go into trials, they often do pretty well, stabilize whatever, when they're in a trial and being supervised pretty closely. So in both groups, those on rituximab and those who were on the placebo, they basically both were able to reduce prednisone 75% of their baseline amount to a similar degree. It was 56% of the patients on placebo and 60% on rituximab. That's hardly different, and it wasn't statistically different. And so it showed futility. And even when they looked at secondary outcomes, the rituximab patients maybe did a little bit better on these different outcomes we use in MG, but it wasn't statistically significant. Unfortunate, but that's where we are right now with the most formal trial of rituximab to date. And I don't know of, they're looking, as Dr. Sukdev mentioned, they're looking at anti-CD38 type agents and maybe some CD19 agents will be looked at in MG, but that it was a little bit of a damper. Not that rituximab is not used anymore in MG, but you know, there was always hope we'd have a positive formal trial and go forward to a phase 3 study. What I just showed you was a phase 2 study. That's what that NeuroNext NIH network does. All right, so we're going to leave the B cells behind and we're going to skip over here to the red, to the complement. And I just want to show this picture and just concentrate on this because Dr. Sukdev described this nicely, but just a little bit more detail. What eculizumab and these other C5 agents like zilucoplan do is they prevent formation of C5b, which is green here. And C5b is that complement component that first allows formation of this yellow blobby thing, which is called the membrane attack complex. And it's named well because what it does is when it's there, it attacks membranes and puts holes in them. And so normally, why do we have this to begin with? It's part of our innate immune system. It's there to push holes in bacteria and other invading things that will cause infection in us. And so that's why it's there. But in immune-mediated disease, it can cause problems. And here it's actually pouring holes in the muscle membrane. Here's our nerve up here at the top of the figure. Here's our muscle again. And you can see what it's done in this schematic. These are the nice folds that are normally present on healthy muscle. But after you activate complement and lead to the MAC formation, which is C5b through C9 of the different complements, there's a lot of different complement molecules. Anyway, you can see that it's simplified. It's destroyed the muscle membrane. It's simplified these nice folds so you don't have a proper morphology, a proper formation of the postsynaptic membrane with nice, densely arranged acetylcholine receptors and so forth. So that's what these drugs do, is they prevent formation of this so you can go back to a normal state. All right, so the REGAIN study got eculizumab approved. I don't want to go through too much of the statistics and the worst rank analysis, but anyway, most of the outcome analyses, besides the pre-specified primary one, but all these other analyses that were pre-planned showed that the drug worked and it got approved, okay? And this is some of the data that I'll show you just to give you an idea. In red is placebo for these different outcome measures. For instance, this is the MG-ADL that some of you may have done to yourself on yourself. It's an easy 8-item questionnaire. Here's the QMG, I believe, yeah. And here's the MG composite. Anyway, you can see very quickly after just a few weeks, a reduction in score. Reducing the score in these scales is good. It's good. The eculizumab does significantly better than placebo. And when the placebo patients cross over to open label eculizumab, okay, their scores come down, just like we saw initially with those who you could say were lucky enough to be randomized to the eculizumab initially. So you again see a nice response over a period of weeks and realize the data here is blinded, and so is this data. The investigators who were evaluating the patients had no idea what the patients were on here. They didn't know where the patients were coming from as far as their treatment experience, okay? And then it became an open label type process. Okay. The zilucoplan was mentioned in the prior talk. So eculizumab has to be given intravenously. The least frequent that we know to give it is after that first 5 weeks, you give it every 2 weeks, but it's still every 2 weeks getting hooked up to an IV. Not the easiest, not impossible, not the easiest thing to do. Infusions can be done in a variety of settings. But anyway, this is not an antibody, zilucoplan. It is a macrocyclic peptide, okay, as Dr. Sakhtov mentioned. But it basically does the same type of thing. It prevents C5 from splitting into C5a and C5b. This is given every day with just a quick sub-Q shot. It's still under investigation, obviously, but phase 3 studies are underway. But let me show you some of the data. I'm not going to show you data for the lower dose. I'm only going to show you data for the 0.3 milligrams per kilogram dose. Like the eculizumab trial, these were tough patients. Look at the QMG score. This was 18 to 19 for the different arms. I can tell you the thymectomy trial was 12. Again, the lower the number, the better you're doing, the less severe your MG. These were pretty severe MG patients who had MG for a long time who were enrolled, 8 years on average. Okay, here's the data. Again, the lower you are on these graphs, the better as far as your score. And you can see the zilucoplan versus the placebo in gray, both for the QMG and the MG-ADL showed significant improvement. It was a P of 0.05. That's right at the threshold, but these were small phase 2 studies. Okay, this is just another way to look at it, the proportion of patients who had an MG-ADL score that was perfect, 0, or just a 1 out of 20, a possibility of 24, doing very well. And you can see on active drug, it was much more likely to have this minimum symptom expression than placebo. How did patients do as far as side effects? They really did quite well. Patients with related serious adverse events, none were seen on any of these 3 treatment arms. You can see here, I have it in red. Patients requiring rescue at the higher dose of zilucoplan during the trial was 0. None of them needed it, okay? Only 1 on the lower dose, whereas 3 placebo patients, a fifth of the patients on placebo, had to have some rescue therapy during the trial, okay? Yeah, there was some injection site bruising. It does occur. This is injected, a scab here and there and so forth, but pretty well tolerated. Okay, now I want to mention this because it's important. You cannot, again, you cannot take these drugs without getting immunized for Neisseria meningitis. That's also known as a meningococcal form of meningitis. Meningitis is potentially deadly. This meningitis kills college students. Shouldn't anymore, as long as the college students are getting immunized before they go off and leave home. And most colleges require it. My kids got it. I didn't. I'm old, but my kids were immunized for this. There are 5 different serogroups that we have vaccines for. The ACWY is the quadrivalent. Quad is 4. I think you all know that. There's 2 forms of it. You have to take initial doses, and then there's typically a booster a few years later. The B, those cannot be substituted between the 2 different vaccines. There's initial doses of 2 or 3 doses in the first few months. And then at least for the Trememba, you have to have a booster at a year. You've got to do both of these. It's really important. You need to be immunized 2 weeks before you go into the trial. And do realize it doesn't make it 0 as far as the possibility of getting meningitis. So in the longer term data with Soliris, with eculizumab, 1 patient did develop meningococcal meningitis, but they did fine. They did fine with the antibiotics, okay? But anyway, it does significantly, basically, it almost reduces to 0 your chance of meningitis. It just doesn't make it 0, okay? And why? Because complement is important in fighting off this particular type of bacteria. It's an encapsulated bacteria, and complement is important for that. And we're blocking complement with the drug we're using to treat your myasthenia. Okay, FcRns. I have 10 minutes. Ugh, sorry about that. FcRns, just a very quick schematic. This is how they work, okay? So normally, these are IgG, these greenish, bluish things. They normally are taken up by cells, endothelial cells, and they bind to this red and yellow thing, which is the FcRn molecule, and they do that in endosomes. But when they bind to them, then they will get recycled back out into the bloodstream, okay? If you have a molecule that blocks that interaction of the immunoglobulin with the FcRn, they can't bind to the FcRn, and they are then going to be destroyed, basically. They get degraded in lysosomes if they're unbound. They don't get recycled. That's how we reduce IgG. There was a question that was asked, is it only the antibody for myasthenia gravis that gets degraded? No. It's across the board for IgG1, IgG2, IgG3, IgG4. Not so much for the other IgG types, but anyway. But that's why this could have broad applicability, because it covers not just IgG1 mediated disease, but potentially IgG4 and so forth. And it reduces IgG by about 70%. It's very similar to plasma exchange as far as a reduction. Okay. And so these are potential advantages. I want to show you just a couple pieces of data real quick. This is efgartigimod, which is the one that's most studied, and the phase 3 study is done now, and I have that data, although it's really just a press release so far. But this was the phase 2 study. This is given weekly in an infusion for 4 weeks. That's how it's mostly been looked at, but you can repeat those cycles as needed for 4 weekly cycles, okay? And it was well tolerated. Headaches do occur with this branch of drugs. Like with a lot of infused drugs, you can't get headaches. There was only one case of a herpes zoster on the efgartigimod arm. That patient, by the way, was also on prednisone and mycophenolate. But anyway, here's the data that I just want to show you. Again, down is better. And you can see for the different outcome measures, and every time there's an asterisk here, that means it's actually a significant p-value, but the patients who got efgartigimod did better than those on placebo, which are in the black line here, okay, on these different scales. And this is just another way. This is looking at responder analyses, the proportion of patients who had a given point reduction on these different scales. And what you can see here, at day 29 and day 36 for the MG-ADL, you can see the green bars are wider. You had more patients meeting that certain point reduction with the active drug than you did with the placebo, okay? Here's the phase 3 ADAPT trial data that really just came out a few months ago. And so again, weekly 10 milligrams per kilogram IV dosing. And you can see for the MG-ADL, the QMG, and minimal symptom expression, that MG-ADL score of 0 to 1, the proportion of patients who met that with efgartigimod was significantly higher. Look at those p-values of less than 0.0001 than placebo. And again, well tolerated. Headaches were 28% in both groups, whether you got placebo or efgartigimod. There were actually fewer severe adverse events on the active treatment. UCB, now we've had industry representatives from just about every one of these companies on today, which is great. They're our partners. We need them to do these types of trials. The NIH doesn't have money for every single clinical trial. They just don't. There's not enough money. We need industry involved with our trials, okay? They're crucial partners. This is UCB's form of anti-FcRn therapy. It's an IgG4. It's not an IgG1. It's an IgG4 monoclonal antibody. But again, it will reduce all forms of IgG through that mechanism I showed before. It was somewhat complicated. I don't want to get into too much detail, but we need phase 3 data with this agent. But again, there is a signal here that it does help. And you can see the separation. This is based on the QMG here. Again, the lower you are on that graph, the better you're doing versus a placebo. And then they switch patients over to different doses after the initial 28 days. But there is signal here. This mechanism with this agent is working as well. And then finally, nipocalimab, which is another IgG1. And they looked at a variety of different doses over an 8-week study versus placebo, okay, at different doses per kilogram. And when they put all those different doses together for the active arms, 52% of patients had an MG-ADL reduction of at least 2 points, which is a good, well-respected drop in that MG-ADL score, okay, versus just 15% on placebo. Again, a significant p-value of less than 0.05. It was 0.017, okay? And again, these agents seem to work pretty fast. I made that point with the crisis blurb that I mentioned earlier during the sessions. It works pretty fast. Maybe these are ways we could deal with crisis. We'll see. Again, you don't need to have an immunization beforehand. It can work across all different MG populations. They seem to work fast. Plasma exchange is tough to give. It's not available in all hospital systems. IVIG is another standard we use for crisis. There's sometimes shortages of IVIG. It is a blood product. which has concerns, although the supply is quite safe. But anyway, could this be another way to deal with acute exacerbations? Because it works fast. Okay. And this is just a summary of these different agents. The first four here are FcRns. Ravulizumab was already mentioned, which would be a way to give an intravenous infusion for complement. For C5 inhibition less frequently, it's every 8 weeks instead of every 2 weeks, and zilucoplan is just a sub-Q daily. And you can see these agents are being looked at in other diseases besides MG. Here's CIDP, IMNM, what the hell is that? Immune-mediated necrotizing myopathy. Okay, all right. I am gonna skip comments about thymectomy. I'll answer questions about it. I'll just say that the extension study did show continued benefit even at 5 years after the procedure over prednisone alone, and we published that just last year in Lancet Neurology. And this is based on the QMG. Again, the lower you are on the scale, the better you are. The QMG values were better, and the prednisone requirements were better. And the typical patient after 5 years who had thymectomy was just on about 10 milligrams every other day of prednisone, a pretty low dose. All right, and just more data from the extension study. I'm gonna finish up very quickly. We have treatment guidelines that were published in 2016. This is an international panel that Don Sanders and I have co-chaired with Dr. Narayana Swamy, who's at the Harvard system, acting as our facilitator, and she is our expert in guideline production and also using the RAND UCLA methodology. And we do have a patient representative through the Myasthenia Gravis Foundation of America, and we just added for the 2020 update my colleague at the University of Chile, so we'd have a South American in addition to our Asian and our European colleagues. Anyway, these were the initial categories that we looked at in 2016. This is what we're looking at for the update that we started last year. It's going to get published hopefully later this year. Ocular MG, thymectomy, methotrexate, rituximab, eculizumab. We had put in some physical training, but unfortunately I don't think that's going to make the final cut. And then also some comments on checkpoint inhibitors, which are used primarily for cancer treatment, melanoma, lung cancers, other types of cancers. But those can create problems in regards to myasthenia gravis. And we have a few comments about that in this. And with that, I'll finish, and I'm happy to try to answer some questions. I hope everybody heard me. I hope I wasn't talking to myself.
It's Tony, Gil. I was listening to you.
All right. Hey, Tony. Hey, were you on for any of the ICNMD stuff? I'm sorry, everybody, but I'm talking to Tony.
I made sure that I got connected about 15 minutes ago. So
okay. I was talking about the Spanish meeting that is running from 3 in the morning to about 8 in the morning. Anyway. Okay. Questions. Let's see what we have here.
Okay. So you mentioned about a lab getting a positive AChR for previously negative patients using a more sensitive clustering technique. What was the name of that test?
Oh, of the clustered, it's just called the cell-based assay, but it's not commercially available.
Okay.
So you can get them. Tony, is anybody in the U.S. doing them now?
Not that I'm aware of.
All right, so you can get them in Greece, and you can get them in Oxford. I think there may be some place you can get them in the U.S. These are labs that are doing them. These are academic labs, university-based labs. They will do them. They will charge for them usually. It's hard to get them ordered. I'm hoping they will become commercially available at some point because, again, there are quite a few seronegatives out there who probably do have receptor antibodies. And once they'll show up on the cell-based assay, it will really, first of all, affirm the diagnosis and help us better manage the patients. But they're just called cell-based assays, Okay. But they can do them for MuSK. They can do them for receptor antibodies. They can do them for LRP4 as well. When I order them, I get all three. They just run all three for me at the Oxford lab. So,
okay. This person is asking, why aren't follow-up comparison tests run? They were diagnosed a couple years ago, and they have a binding number and an AChR modulating number, but they
Yeah.
haven't been retested. Why is that?
Well, I don't believe in retesting in most patients.
Okay.
In studies, we do them. This is why. All right. And a question came up before that I don't think I answered yet. Does prednisone lower your antibody levels? Yes. Most things we do in MG that are therapeutic, mestinon wouldn't, but just about everything else, at least for a while, will lower antibody levels. In plasma exchange, they will lower the antibody levels. Most of these drugs that we use, the oral drugs, they lower antibody levels. All right, so this is a deal with the antibody levels. Usually an MG patient won't improve unless their antibody level goes down. But you will see patients who haven't improved whose antibody levels go down also. They really, and Duke has done a huge analysis. They have a huge database at Duke University of MG patients. They've done several studies on this. The antibody levels, even though we wish that they would predict treatment response, how patients are doing and so forth, they really just don't do it very well. Maybe Musk antibody levels do it a little bit, but it's just not perfect. And so I generally don't reorder antibody levels, okay? And that's why there's just isn't a great reason to do it.
Are there any side effects or problems caused by being on 2,500 milligrams or greater of, is it mycophenolate?
Yeah, it'll be mycophenolate.
For a number of years?
For that dose, the only thing I could think of at that high a dose is mycophenolate. Methotrexate, tacrolimus. Yes.
There's some long therapy names.
Right. It's mycophenolate mofetil, and it's also known as Cellcept. And there's also mycophenolic acid, which is slightly lower doses in milligrams, but it's the same basic thing. Okay, so you can get side effects at any dose of just about any of these things, but typically the side effects are greater at higher doses. Mycophenolate, a lot of us like using it because it doesn't cause major organ toxicity, we say, meaning unlike azathioprine and methotrexate, you generally don't get liver problems, you don't get kidney problems with it. But it sometimes can cause some GI disturbance. The most common side effect is diarrhea. It also, the reason you should be getting your CBCs checked regularly, I'm not saying every week, but every 3 to 4 months is eventually what I do with mycophenolate patients who use mycophenolate. It can cause leukopenia. It can sometimes cause low platelets. That needs to be monitored. That's true at 2,500 milligram doses. It's true at 1,000 milligram doses, okay? You can see problems. So yes, it can cause side effects. I've seen viral reactivation syndromes with CMV with mycophenolate. These are rare. They shouldn't scare people from using them, but you just need to be potentially aware. Just like if you need eculizumab because your disease is so badly controlled, you shouldn't be running around worried about getting meningitis, all right? Because the risk is really quite low, okay? There are gonna be problems. There are gonna be problems with the COVID-19 vaccine. There will be with the COVID-2 vaccine. Somebody's gonna have a problem, but it's gonna be a rare chance. And so I don't think most of us walk around, at least when it's not thundering above us, that we're gonna get hit by lightning. That's the best parallel I can give you. All
as
right.
it's thundering where I'm at right now.
We have sun.
Would being on Cellcept affect a CBA test result?
Would being on Cellcept affect a CBA result? But it's potential. So these cell-based assays, they're picking up somewhat lower affinity antibodies is what they talk about. If you reduce the number of those antibodies because of the therapy that you're on, so you have fewer of those low affinity antibodies, I guess it could. I think that would be the case, but they are quite sensitive. I think it would be unlikely, but possible. Yeah.
Okay.
Look, it's pretty rare with the standard immunosuppression. It does happen, but it's pretty rare in a myasthenic who is on therapy that their antibody levels go to 0. It does happen sometimes. I've seen it happen, but usually there's still a very small titer there. Even though you're doing great, you feel normal, but your antibody levels have come down nicely if they have been retested. And again, I'm not advocating retesting in most situations, but there is still some antibody level there in most people. So I think it would be unlikely. Okay.
Okay. Which labs are proven to provide accurate lab results in testing for ABs besides
Oh, boy.
AChR and MUS?
Right. I can just tell you, this is getting into, I can just tell you where we send ours through. We send ours through the Mayo system. I have that set up here.
Okay.
I trust their clinic, their clinical laboratory. Yeah. So when I get the binding and the modulating together, it's going through them. Yeah.
Okay.
As long as insurance allows it. All right.
Okay.
All right.
I could probably take one more question. If zilucoplan is a peptide, can it get removed by Plex?
A peptide is a protein. Plasma exchange removes just about every protein. That's why we give albumin back sometimes. I would think it would, yes, just like it would an antibody. Yes, yes.
Okay. We've got a couple more questions here. Let me see.
And I'm happy for any of the other speakers to, and if they have if they think I said something misleading, but I think it would remove.
This person just said you answered my gastrointestinal problem.
Oh.
Another person just typed in, so once you're diagnosed with MG, you'll always have MG.
So that's generally what I tell people. Is that 100% true? There are cures of MG. I think thymectomy helps us do that. It doesn't happen all that often. I like to think more of remission, and we love to get patients in remission, and I wish it was 100% of patients go into remission. But anyway, once you have MG, for the most part, yes, you always have MG, but it could be controlled very, very, very well. All right.
Okay. All right. Well, thank you very much. We are going to get set up with Dr. Amato next. So I appreciate your time this Saturday, and hopefully, I don't know if you're going to get a cat nap in before 3 o'clock or not.
I think it would be dangerous.
You might not get back up.
I've got
All
to go
right.
to your birthday party. Hey, everybody, I hope you enjoy the rest of the concert, the conference. I'm going to listen to Dr. Amato. And exercise. Dr. Hafer-Macco is going to give a nice talk about exercise. Exercise is almost always good. It's good. Listen to her. Okay, good.
Perfect. Thank you.